We may soon be able to target more of the proteins that we once called undruggable. Molecular glues have already changed how we think about druggability. They work by sticking to the surface of a degradation-inducing protein like cereblon, creating a compatible surface for target proteins and luring them to their death. No drug binding pocket required. But even molecular glues have limits. They mostly recruit proteins with a particular kind of loop structure present on about 7% of proteins. Or so we thought. This paper shows that that loop is actually not a strict requirement for molecular glue targets, opening up a potentially vast pool of new targets for degradation and thus possibilities for disease treatment. The team takes two main approaches to identify potential new target proteins. First, they mine the proteome for a minimal sequence of amino acids that appears to be important for engaging the cereblon molecular glue complex, yielding a new structural feature capable of binding cereblon. Second, they search for proteins with surface patches resembling those of known molecular glue targets. And they find some. Interestingly, these proteins don't necessarily structurally resemble the known molecular glue target, they just share a similar patch of surface. It's really cool that they can even find that. Their hits include VAV1, which is of interest for treating autoimmune diseases. So they apply a compound library to see if any drugs can bring it into contact with cereblon. Indeed, one compound does, and, well, the company is actively developing VAV1 degraders. So clearly there's a lot more to be learned about which proteins are viable molecular glue targets, which is really exciting, especially considering that leveraging other degradation complexes or applying different compounds could open up even more possibilities. So this will be an exciting space to watch.